Hello and welcome to the Light-Sheet Chronicles. I'm Dr. Elisabeth Kugler, a science communicator and your host of today's episode, Imaging Across Scales, from subcellular structures to whole organisms. Light-sheet fluorescence microscopy is a cutting-edge imaging technique that combines speed, precision, and minimal light exposure to capture intricate biological details. And in today's episode, we will talk about light-sheet fluorescence microscopy to image across scales, applications, and the whole world of tissue clearing. Today, we have three wonderful guests. The first two are from Gubra, a company that specializes on preclinical drug development. And our first guest is Dr. Urmas Roostalu, Senior Department Manager of 3D Imaging in Biotech and Drug Discovery. Hello, Urmas.
Hello.
It's great to have you. And we have also Dr. Johanna Perens, Specialist in 3D Imaging and Image Analysis. Hi, Hannah.
Hello.
It's great to have you. And then lastly, we have from Bruker Luxendo, the Light-Sheet division of Bruker, Dr. Malte Wachsmuth, the Managing Director of the Luxendo GmbH. Hi, Malte.
Hello.
It's great to have you all here today. And so what we're going to be talking about is imaging across scales, because one really great aspect about light-sheet fluorescence microscopy, and which makes it so unique, is that we can perform imaging from subcellular structures to whole organisms. And so not only can we span this large range in terms of sample size with light-sheet microscopy or SPIM, but we can also, on the one hand, image really fast sample dynamics in vivo. And then on the other hand, we can image really large cleared samples. And so before we talk about large and cleared samples, I would love to start a conversation with in vivo imaging and to study dynamics especially. And so, Malte, maybe you could share with us some typical in vivo imaging experiments and talk about the speed and requirements that are needed for such experiments.
Yeah, of course. I mean, I think the most typical, almost classical in vivo imaging experiment for which light-sheet microscopy has, I dare say, been developed is early development of, for example, Drosophila embryos. So what people want to do when looking at early Drosophila embryonic development is to follow individual cells, individual nuclei from very early on and follow a bunch of division cycles and then see how cells differentiate, how they turn into wing discs, into different organ predecessors from the very homogeneous initial state they come from. And in order to be able to do that, you have to be able to image a whole Drosophila embryo, which has a diameter of, let's say, 200 micrometers and a length of 500, 600 micrometers in total, but with a very high time resolution. So you must be able to see every individual nucleus of every cell. And let's say it's 1,000. And you must also be able to image the whole object, the whole embryo fast enough so that you can basically track every individual cell division. These cell divisions occur basically in a very synchronized way. So they almost happen simultaneously across the whole embryo, a mechanism that is part of the research basically. Why is that so? How does the communication work? And in order to not lose any of these nuclei, then you really have to have a very high resolution. For example, you must be able to image the whole embryo every 30 seconds, and this for maybe 20 hours. Of course, when you acquire images from a fluorescently labeled sample, you have a lot of photo bleaching. So you need a method that basically doesn't bleach at all. So you can track individual nuclei for 24 hours every 30 seconds. And this is where light sheet microscopy really excels because it's very gentle to the samples. If you keep it low enough, it really doesn't introduce any photo bleaching. And yet you can basically image fast enough and with a resolution really in the range of a micrometer so that you can track individual nuclei for such a long time and without losing individual nuclei and without bleaching them away. And this has been proven 10, 15 years ago with a couple of seminal papers. And I think what is really fascinating is that you can see that you can follow 10, 15 division cycles without losing individual individual nuclei. Then maybe on the other end of dynamics, this sounds already fast, but it's a very large object. And also what drives basically the differentiation of individual cells, individual nuclei to become a certain organ, become part of the wing disc, for example. This is among many other mechanisms driven by molecules called morphogens that basically control the differentiation of individual cells. And another question one would want to address with light sheet microscopy is, how do these morphogens distribute? Do they develop gradients within an embryo? Do they diffuse, or are they somehow controlled by binding to cellular or nuclear structures? Probably nuclear structures serving as transcription factors. And this then guides us to another modality that you can use with light sheet microscopy, and that's really imaging very fast, for example, in a single plane, so that you can also track the movement of individual molecules or individual complexes. For that purpose, of course, you must be able to image with really a time resolution in the millisecond range, which allows really to characterize quantitatively the dynamics and the mobility and also the binding of individual molecules to nuclear structures. And this helped to really demonstrate that indeed when you look at such an embryo, you really have a gradient of these morphogens, and the gradient of these morphogens basically determines, or the concentration of different morphogens determines the differentiation goal, or the fate of individual cells. And that's basically how a spatial differentiation is controlled in an embryo. And light sheet microscopy is probably the only fluorescence microscopy method that allows to, on one hand, really follow the whole embryo at once with a good time resolution, but also follow individual molecules at the same time within this embryo. And this is really a big strength of light sheet microscopy to many other microscopy methods.
And thank you for mentioning morphogens in the first 5 minutes, which is amazing, because as biologists, you spoke now a lot about these long time lapses where we look at hours to days of development. As an example, you used Drosophila and wing disc. But for example, zebrafish is another very typical experiment that we look at. And so we looked at these long time lapses, but I also know that we use light sheet to image very fast dynamics. And you very briefly kind of touched on it, but maybe you can give us another example, such as, for example, calcium imaging or red blood cells.
Yeah, at the end, what counts is time resolution, is speed for these applications. Very often you do not then image an entire object or an entire 3D sample, but you restrict yourself to either a single plane or a few planes. Like this, you can really get to a time resolution in the range of milliseconds. And with the sensitivity light sheet microscopy provides, basically using high sensitivity cameras and really a very, at the end of the day, minimalistic optical setup, this really allows to be fast, be sensitive, and be also very precise in terms of spatial resolution at the same time, which you need for this type of applications.
Awesome. Because we also now come to Johanna. Because in the first episode, we spoke a lot about large cleared samples, which is what you do at Gubra. But I think it would be nice to just come back to general image analysis challenges, because in working with large cleared samples and in vivo imaging, you always have really large data data samples that you have to work with, and obviously this huge data load is a bottleneck, and you have various different challenges. So can you maybe in general speak to the challenges in image analysis with light sheet microscopy?
As you mentioned, there are quite a few challenges that come with analyzing light sheet data. And in the first episode, we also talked about the problems of having a lot of data and how to handle it. But I would like to this time maybe go more in depth or being more specific about some other challenges that we also have additionally. So, for example, imaging process itself introduces various artifacts. For example, you can get them from clearing process, or the reflections, possibly from the sample, or even cuvette or so on, or uneven illuminations across the samples. So all these can affect how clearly we see and interpret the structures. Then another challenge that we have been facing is, for example, a chromatic shift. So So chromatic shift means that different fluorescence channels don't quite line up due to the optics, meaning that resulting 3D volumes need to be shift corrected later on during the post-processing. They need to be corrected very carefully, especially, for example, if you would like to do co-localization studies where precision is really everything. On top of that, it's very important to be vigilant about the stability of the whole setup. So, for example, microscope has a drift over time and also small changes in how tissue is processed. So depending, for example, what protocol you use or what you would like to image. So that can also slowly shift your results over time. And if you don't catch these changes early, you can start noticing that, for example, your segmentation models, classic or AI-based, they can start underperforming, actually. So it is very important to have systems in place to monitor these variables closely. And so for that, quality control is the key. It's an essential step. At Gubra, for example, we spend a lot of time checking the quality of the scans, of segmentations, or also, for example, for brains, you do atlas registration. You also need to be sure that the registration is done really well. So we're always pushing to automate also these steps as much as possible so we can scale while still trusting our results.
That's amazing. You're mentioning so many important steps, from sample preparation to automation to quality control, and also that there can be quite a lot done actually in post-processing. So I think it's important to mention that all of these individual steps can really contribute to good data, but unfortunately also to bad data. So it's really important to know your microscope and your samples as well. So maybe before we kind of move on to tissue clearing, I'll come back to you, Malte, and ask you a little bit about in vivo imaging again, and about sample tracking and what you have for sample tracking in light sheet microscopy.
So sample tracking is actually a functionality of a microscope that allows you to follow an object, a living object, as it develops, as it grows, as it migrates through your sample holder. This is something we observe quite often, for example, for embryo, when imaging embryonic development. Obviously, the embryo is developing, is growing, and is also moving a little bit. And what we do is we basically capture the image, analyze a time point, a whole 3D stack, and then can correct the position of the stack for the next time point so that we still acquire the whole object. This can be applied to every time point. This can be applied to every fifth time point. And this is a very efficient way to make sure you do not lose your object, especially for long-term observations. If you want to image the development of an embryo for a day or for 2 days, then you certainly do not want to sit in front of the microscope and do this by hand. And this work, the way we do this is based on image correlation. That means we kind of compare the similarity of the structures in 2 subsequent time points, and we try to make this overlap as well as possible. That's effectively what this algorithm does. And like this, we have a very robust way, even for objects that grow and maybe become a little bit too big for a single field of view. And at least conceptually, this also allows to identify such an event and to change, for example, the zoom level of the microscope, so that when you detect that the object grows so big that it cannot be captured within a single field of view, you can either take what is called a tiled acquisition, or you can zoom out and basically image with a larger field of view. That's all functionality that can be implemented and is to some extent already implemented in our software.
Yeah, which is amazing because, like you say, sometimes a sample might grow, like for example a zebrafish tailfin can grow out of the field of view, but I know you also have some examples of actually plant roots growing out of the field of view, and I think that's really cool because you don't initially think about, for example, plant root imaging. So I think it's a very cool example. And moving on now from in vivo imaging, we kind of want to talk about tissue clearing a little bit, and that brings me to you, Urmas, because we know that imaging across scales is really interesting if you want to understand biological function and compound distribution in whole organs and whole organisms. And this normally requires fixed and clear samples. So maybe, Urmas, you can talk with us a little bit more about what tissue clearing is in general and why it is important.
Yeah, absolutely. I think you, Malte, and you gave a very nice introduction into developmental biology and the role imaging plays there. But when we want to move into larger samples, we cannot really see through them. The light doesn't pass them. And tissue clearing in essence means making the tissue transparent so that we can shine a light through it and actually visualize everything that is deep in the tissue, really in the center of a large mouse organ or a human tissue biopsy. And if we consider a typical organ from a mouse or a human tissue, then the light penetration is impaired because all the building blocks in the tissue, the individual cells, the proteins, they have different refractive indexes. And this really scatters the light around, so we cannot really shine a light in a single line through it. Plus, there are also all the pigments, whether it is the melanin in the skin or some other pigments in other tissue types. So all this inhibits imaging in a large scale. So if we want to move into really centimeter wide tissue samples, then we need to make it transparent. And there are many different methods developed over the years, and in large they can be divided into two. So they are either aqueous tissue clearing methods or organic solvent based. But what they all have in common is that they try to get rid of the pigments in the tissue and match the refractive index of these individual components. So finally, when we end up in imaging, then we have this highly transparent tissue, and we can visualize individual fluorescent cells very deep in the mouse or rat organ or other large tissue samples. So in our team, we primarily use organic solvents to make very large tissue samples transparent. And this then opens up a large variety of downstream applications. So we can carry out antibody stainings to visualize many proteins in the same sample. We can visualize mRNA molecules and also fluorescently conjugated drugs that we then study in preclinical research. And then downstream, we can use all these methods in both in basic biology research, but also in drug discovery. And for us, what is very important, then it opens up possibilities then to study drug biodistribution, the efficacy of drugs, and the mode of action of drugs, how actually novel drug candidates work, to identify actually which ones are worthwhile to take further in clinical development.
Which is incredibly important, especially if you work preclinical, you really want to know the most about the drugs before they would go into any clinical trials. But as you mentioned, there's many, many aspects to drug discovery, and obviously you guys do this a lot and you have the inside expertise. So I was really curious if you could share with us, some more information about the general setup and the challenges that you work with in drug discovery.
Yeah, absolutely. I think in the end, what we have learned over the years is that there are two main aspects of it, right? So one we already a little bit touched upon was the tissue clearing. So how to customize that to different tissue types we are working with. And for this reason, we really try and work with different organs. So very small ones, so isolated aortas from preclinical animal models to really large ones like whole rat brains and kidneys and skeletal muscle tissue types. And all these have the different clearing requirements. And also, for example, when you take into account the bones and the spinal cords, they require additional clearing steps to make the bone tissue transparent. And overall, when we consider preclinical imaging, there is one underlying principle that we really emphasize, and that is the precision. So we need to be absolutely certain that the conclusions what we take on a new drug candidate, they are valid. And this, of course, because the drug development process is relatively expensive and time consuming. So every time we make a decision, it actually carries significant impact. And to get to this precision, of course, what Johanna has a little bit discussed is image analysis, but overall it relies on large sample sizes and throughput. So having imaging from a few brains is not enough. So we need to take it actually to tens or hundreds of samples to make the conclusions that actually valid, have a good statistical significance to make the right conclusions. So everything we do at Gubra is really geared towards throughput. So we have build custom-made robots that take care of the sample preparation steps that can actually stain hundreds of samples at the same time and clear them. And then we have also, working together with Bruker, we have developed and optimized commercial microscopes for high throughput imaging, so that we can load and image hundreds of samples one after another in a very kind of like unified environment in large batches. So we also minimize the batch dependence. And so everything is really aimed for throughput, and that of course have all the consequences of imaging and image analysis that Johanna has mentioned. But there are many different opportunities that it also brings with it.
It sounds like a really futuristic lab, though. Now I'm like really imagining, loads of robots and like loads of samples. So I guess it's lots of fun to work there.
Absolutely, you're absolutely correct. And also the robot part, it's also brings its own fun to have these robots that you can imagine in a car industry handling tiny tissue samples like brains and putting them into the microscope and doing this one after another. But we also learn a lot, and that is maybe the little bit like a take-home message. What we have figured out by 3D imaging is that it kind of like opens up so many new doors. Like with the traditional histology, maybe we look at one part of a brain and make our conclusions, but that conclusion maybe is not valid for the entire organ. For example, we use this technology a lot in neuronal activation imaging, looking at where a drug reaches a brain and where a drug activates neurons. And every mouse brain can be divided into hundreds of different brain regions with functions. Some of them we know, some of them we do not. And then we can start to make conclusions and predictions on what is the effect of a drug in a mouse brain. Does it activate parts of a brain that regulate appetite, which may be good for a weight loss drug, but maybe it also leads to excessive nausea and headache or a migraine-like side effects, which we then want to avoid in further drug development. So all of these things that you would never imagine getting from looking at particular mouse brain region at the time. So you really need to have this holistic picture of the entire organ. And the same is true when we look at the efficacy of drugs, how well they work. So we are just now working on a heart failure project. So mouse has a heart failure similar to a human, triggered by high blood pressure, kidney failure, diabetes. And then we start to look at where a drug has an effect in the heart and whether the effect is beneficial or not. And then we actually used a small molecule dye to label fibrosis to characterize in a whole organ level how much the drug reduces fibrosis and where in the heart. And the heart, similar to the brain, we can divide into all these different anatomical regions. And then looking at the entire heart, and we can see the effect of a drug along blood vessels on this kind of like a perivascular fibrosis, which is of course very good like endpoint, because perivascular fibrosis has a significant pathological consequence on cardiac function. And these are just a few examples, but it's really this whole organ imaging that light-sheet microscopy and tissue clearing brings with it. This is what opens up all these opportunities. And maybe another example is a kidney research, right? We are working quite a bit also on polycystic kidney disease in imaging, which is a horrible disease, like causes this massive fluid-filled cysts to develop in the kidney. And they also develop in a random pattern, and there are tens or hundreds of them depending on the stage of the disease. And then we can use this 3D imaging to look at the entire kidney and see where the drug actually has an effect. Does it reduce all the cysts? Does it reduce the cyst formation or further growth? And then we can use this kind of imaging to characterize it further.
It's so interesting. I could listen to you for ages, to be honest. I think especially also what you mentioned, you, for example, look at the brain, but then also at the heart, and for example, also at the kidneys. And what I think is really interesting is that you mentioned as an example perivascular fibrosis, which is just one example as an endpoint. But I think what is also interesting is that you can take different organs and then compare drug efficacy in different organs, right?
Absolutely. I mean, this is in the end what we often begin with is actually in a drug discovery or a drug development process is characterizing first where our drug gets when we inject it. And then we make a drug, we conjugate the fluorophore, and those mice under the skin, which is basically what you would do in a clinic. Like you have a subcutaneous injection of a drug, or maybe also oral formulation, you give it as a small pill, or any other way, you dose it, and then you want to know actually where the drug ends up. Like when we dose under the skin, how much of a drug reaches the cell type you actually want to target with a drug. Is it the cell type in the kidney or a brain or some other cell type? But then what light-sheet imaging actually brings with it is this possibility to look at all these organs, whether we do it in an entire mouse, which is also possible in a maybe a little bit lower throughput scale, or we look at individual kidneys, brains, maybe liver, maybe some other cell types, and then characterize all of them and provide this comprehensive map of drug biodistribution. And then, of course, secondarily, you can start to look at the effect of the drug in all these organs, because of course the drug can have a beneficial effect in one part of the body, but maybe a detrimental effect in another. Because often many drugs, they fail in phase 2 and phase 3 clinical trials because of these unwanted adverse events. And this is exactly what we want to prevent in this kind of preclinical analysis, using imaging, trying to detect these potential events early, and then find out if we can develop a drug to avoid these events, or alternatively then move into a different drug target and drug formulation altogether.
Yeah, it's amazing what we can do nowadays. And like I said, I think your lab sounds really futuristic and like robust. But obviously for Johanna, who is the data analyst in the group today, this is not that great because she has so many data to work with, which is very, very challenging. And so I want to come back to you, Johanna, and say, we spoke now about various kind of scales of data. So Urmas mentioned organ levels, whole organ levels, different organs to compare to, etc. And in the first episode of the podcast, we spoke about data challenges in general. But now, kind of tying onto what Urmas just said, maybe you can speak a little bit about how you compare, for example, data from different organs, because I assume that can be very difficult. And how do you go about that?
Yes, and every new organ we work with tends to bring something new to the table. So either the resolution needs to be higher or the scan area bigger, or which means even more data in both cases. So we always push the limits of what we can process efficiently, actually. And like Urmas explained before about different disease models, so we actually face quite a lot of challenges also by analyzing them. So, for example, if you compare healthy and diseased organs, that can be very tricky. The disease might change the shape of the organ or the way the signals appear. So, for example, our segmentation models need to detect the same type of signal in both healthy and diseased tissue, and to do so with equal accuracy. So to make fair comparisons between the groups. So that's especially difficult in such kind of organs like kidneys and pancreas, or where morphology can vary a lot. Yes, and I would say also that size variation of the objects that we quantify is quite another hurdle. For example, like again, Urmas mentioned about cysts in kidneys. So they can be very big. So big, actually, that they don't even fit into the standard image patch size that we use in AI analysis. So because it needs to also fit into the GPUs we use. And we already use quite powerful GPUs. So, and here we are really limited to this GPU memory. So there is only so much we can do in one go. Another complication comes from the biology itself. So sometimes the cells we are trying to detect are very sparse. And this makes actually very tricky to find data to do your AI model training. And in other times, the cells or the signals might be so densely packed that it's very difficult to distinguish individual cells, for example, between each other. So it's And like I have also mentioned, again, when these same cells, if they're stained faintly in some regions compared to others, that's, again, another source of variation that we need to tackle in our image analysis pipelines. And then there's, because also Urmas mentioned about the brain, and actually, because it is the most complex organ, it's also the most complex organ to analyze in a way. It has many additional steps that we do compared to other organs. So, for example, an additional step is the mapping of sample to standard atlas space so that we can report these signals in specific brain regions. And that obviously requires accurate registration. And I would say this is an active area of research also. So there are classical and also AI-based registration models, but you need to get the brains to map to actually compare the different treatment groups and to good statistical analysis. And if the signals we detect span multiple regions, it sometimes might raise the question, so where should we actually count them? So, I mean, CFO cells are maybe tiny and it's easy to allocate them to a brain region, but what do you do with plaques, for example, that might extend several regions? Do you count them, or do you need to choose a different endpoint that is maybe coverage of a certain brain area? And also there might be, in general, very subtle changes. So local effects inside these brain regions, they actually might get lost when we summarize everything in one region. So then the other option might be a voxel-wise analysis, where you actually do statistical comparisons not at region level, but at voxel level. So actually brain analysis can get complex very, very quickly. Yes. And then just want to mention, there is important part to consider always batch effects. So, for example, if samples were processed or imaged in different batches or separate studies, their signals might differ enough to throw off any comparisons. So we need to be very cautious there. And then we usually, for example, don't compare between different batches of samples.
And I think especially also talking about standard atlas registration is something that I think is really, really interesting, because it relies, like you say, on good data to be there to begin with. But then also this question of what scale of analysis do you choose? Do you look at voxels? Do you look at regions? And I guess, as you say, depending on what drug or, for example, protein you look at, you need to make this decision every single time for an experiment anew, I guess.
Yes, no, that is totally true. And to be honest, very, very often, so because we try to standardize also our pipelines, so it does mean that we do in parallel almost always both region-wise and then voxel-wise analysis. And then sometimes you really might notice that, for example, if you do, let's say, neuronal activity analysis, so you have given a drug treatment to an animal and you would like to see the let's say, neuronal activity response. So this is very local and it's usually very strong. So you have like brain activity hotspots literally in the brain. So it's very easy to distinguish, and they usually also tend to be within the brain regions. So that is quite easy to also visualize, and you can do region-wise, but also voxel-wise analysis. However, there is this problem, for example, if you look at plaque burden or alpha-synuclein accumulation, this is not in every brain in the same place. So they are not in the same voxels, so to say. So voxel-wise analysis does not make sense there at all. So it really depends what's the scientific question, what's the staining you do, what kind of analysis and what kind of endpoint in the end makes sense.
Definitely. And I love that you have this multi-scale approach and looking at it from different angles, which is so, so important. But now I have to move on to a different topic, which is talking about pushing boundaries on a different level. So I want to come back to you, Malte, and talk with you about light-sheet microscopy and tissue clearing, which has been very recently a lot in the media, and everyone is talking about it, which is called the Doritos paper, which is a publication that was published in September 2024. And it has really created waves across the media. And I think many, many labs are at the moment looking at the experimental protocols and trying to re-establish the science. So the paper was authored by the first author, Zihao Oh, in the labs of Bruker and Hong. And in a nutshell, the group used food dye tartrazine, which is also found in Doritos, to make the abdomen of mice transparent. And this is achieved by the so-called Kramers-Kronig relation, where water-soluble dyes can reduce the refractive index contrast between water and lipids, and this leads to optical transparency of live biological tissues. And so this paper was obviously groundbreaking in many, many ways, not only because it uses a food dye to create transparency of tissue, but also because it achieves a non-invasive temporary transparency in living samples. And I know this was like a lot of information now, but maybe Malte, you can share with us why this paper is so important and how this could impact research.
Yeah, I think, like you say, this can really become a game changer. Urmas described this quite well, how tissue clearing works. You basically try to homogenize the refractive index of a sample so that you do not have scattering boundaries, scattering structures, but the light can penetrate on a quite direct path, your sample, which is a really prerequisite for good imaging. And this has been very well established nowadays over the last decade or so, or 20 years for fixed samples, because the treatment to achieve the transparency is usually chemically quite aggressive, so it doesn't really, not really compatible with live living samples. So there's always been the question, if only we could do this in living objects, right? So that we can look into the belly of a mouse, basically, like you said. And I think this paper demonstrates that there is a pathway to achieve this. And I think this paper is also quite fascinating in a different way when you look at it. And I think there's a public domain version available online. So what they basically have figured out that this with this food dye, tartrazine, which is, I think, approved by the European Food Safety Authority to quite high concentration levels, actually. That with this food dye, when you analyze this in more detail, by looking at it from a photophysics point of view, you can see. So if you consider this dye as a kind of an oscillator, which is a bit like an electromagnetic pendulum, if you wish, that you can make oscillate by exciting it with electromagnetic radiation, so light. And when you're close to its resonance, and every pendulum has a resonance, then you don't need much energy to make it oscillate with a quite high amplitude. However, when you go a little bit away from this resonance, for example, higher to higher wavelengths, and smaller frequencies, then you need just much more energy to basically make this oscillate. And the side effect of this more energy required to kick the oscillator is, to stay in the picture, is really that you increase the refractive index in the wavelength range above this resonance. And this tartrazine happens to have a resonance in something around 450 and below nanometers, which is the blue and UV range. And then when you basically look at the solution of tartrazine, you can see that above this wavelength range, at a sufficiently high concentration, the refractive index increases. And this is actually what you need if you want to make a live tissue transparent. You have to basically increase the refractive index of everything that is aqueous, so that you get closer to the refractive index of the scattering structures that mostly consist of lipids and proteins. And this tartrazine actually does this job. So it increases the refractive index of everything that is aqueous, which is cytoplasm and everything in the extracellular space, for example. And the effect is, what they found out here is that it really makes the tissue transparent. It removes all the scattering interfaces, the scattering structures, temporarily. And it's another very strong point, I would say, because it's a food dye. It's also basically digested over time. So it's a temporary way to make a live tissue transparent, and transparent enough so that you can actually do what we usually do with fixed and clear samples, that you can image quite deep into a tissue and follow what basically Urmas and Johanna already talked about, that understanding drug delivery to certain organs in an animal, having some complementary way to maybe follow this in a living animal. I think that's a quite fascinating perspective that this technology, or this kind of technology, may open an avenue to. So this is in many different ways really fascinating, I would say, this paper, and also on a different flavor, in a way that this is based on a collaboration of physicists and of chemists and of biologists, and you need quite some understanding of light-matter interaction to really see that there is potential in it, and then they tried it and it seems to work. So I think if this can be confirmed, this kind of studies and the effect of such mechanisms, this can change a lot and can help us a lot, I think, to make progress in the research we, and especially you guys at Gubra, are actually carrying out.
Absolutely. I'm really excited to see what comes from this discovery. And I think you ended on something really, really nice, which was that we need this interaction and this kind of cross-disciplinary work between biologists, chemists and physicists and many, many other scientists, especially also image analysts, to really make science as fruitful and as groundbreaking as it is today. So yeah, thank you for ending on this. I think this was really, really nice touch, because unfortunately this is all that we have time for today. And so I want to thank our wonderful guests for your time.
Thank you so much.
Thank you.
Thank you so much, guys.
We talked about imaging across scales, from in vivo dynamics and time-lapses to tissue clearing, and gain insights into how to set up tissue clearing experiments, tackle common challenges, and work with large data. So to our listeners, if you want to know more about tissue clearing, make sure to check out the most recent Bruker ebook on tissue clearing principles and techniques, which we will link below for you. So thanks again for tuning in to this episode, the Light-Sheet Chronicles. And in our next episode, From Stopping Hearts to FCCS, we will talk about how to use a laser to study tissue with light-sheet microscopy.
This podcast was sponsored by Bruker GmbH, the Light-Sheet division of Bruker's Fluorescence Microscopy division. Bruker is a manufacturer of scientific instruments for life sciences, materials research, and industrial or applied analysis. If you'd like to learn more, please visit www.bruker.com/light-sheet. This podcast was produced by Keep It Science.